Hello everyone, I am Dr. Keshav Pawar from the Department of Oral and Maxillofacial Surgery, Sri Balaji Dental College and Hospital. Today I am going to tell about sterilization and disinfection in dentistry. Why do we need sterilization? Microorganisms capable of causing infection are constantly present in the external environment and in the human body, and also on the human body. Microorganisms are responsible for contamination and infection. The aim of sterilization is to remove or destroy them from materials or from surfaces which are close to the oral cavity. Sterilization is defined as a process by which an article, surface, or a medium is made free of all microorganisms, either in vegetative or spore form. Disinfection of all pathogens or organisms capable of producing infections but not necessarily spores. All organisms may not be killed, but they can be reduced to a level that is no longer harmful to health. Antiseptics, these are chemical disinfectants which can safely be applied to the living tissues and are used to prevent infection by inhibiting the growth of microorganisms. Asepsis is a technique by which occurrence of infection into an uninfected tissue is prevented. There are physical chemical methods to prevent to sterilize and prevent infection of the microorganisms. Physical methods are sunlight, drying, heat, which is available in both dry and moist forms, and filtration and radiation. Chemical methods include alcohol, aldehydes, phenols, halogens, oxidizing agents, salts, surface active agents, dyes, vapor phase disinfectants. Physical methods, in which sunlight, which it possesses appreciable bactericidal effect due to its content of ultraviolet and heat rays, and under natural conditions its sterilizing power varies according to circumstances, but natural method of sterilization in tanks, rivers and lakes, which is quite effective. Drying is essential for the bacteria to stop its growth. Drying in air has a deleterious effect on many bacteria, but this method is sometimes unreliable and only of a theoretical interest. Heat is the most reliable method of sterilization. The factors influencing sterilization are nature of heat, temperature and time, number of microorganisms present, characteristics of the organisms such as species, strains, and families, type of material from which the organisms have to be eradicated. The mechanism of action depends on whether it is a dry or moist heat. In dry heat, it kills by protein denaturation, oxidative damage, and toxic effects of elevated levels of electrolytes. Moist heat functions by coagulation and denaturing of their enzymes and structural proteins. In dry heat, has different types of methods of heating, which is red heat, flaming, incineration, hot air oven. Red heat is done by flaming through a Bunsen burner till they become red hot. Inoculating wires or loops, tips of forceps, surface of searing spatula, and needles can be sterilized by this method. Flaming is passing the materials through the flame of a Bunsen burner without allowing them to become red hot, just such as glass slides, scalpels, mouths of culture tubes, and incineration is done by reducing the material to ashes. Soil dressings, animal carcasses, bedding, and pathological waste can be incinerated by this. Hot air ovens, which are most widely used for sterilization by dry heat. It is used to process materials which can withstand high temperatures and which are likely to be affected by contact with steam. It is a method of choice for sterilization of glassware, forceps, scissors, scalpels, swab sticks, packed in test tubes. Materials such as oils, jellies, and powders, which are impervious to steam, are sterilized by hot air oven. Hot air oven is electrically heated and is fitted within the small size that maintains the chamber air at a chosen temperature, fitted with a fan that distributes the hot air into the chamber. This is a picture of hot air oven, and it has separate compartments, and the temperature can be set as required. Holding temperature is generally 160 degrees for 2 hours, 170 degrees for 1 hour, and 180 degrees for 30 minutes. Cutting instruments, such as those used in ophthalmic surgeries, should be sterilized at 150 degrees for 2 hours. Oils, glycerol, and dusting powder should be sterilized at a temperature of 150 degrees for about an. The following precautions should be followed. It should not be overloaded. It should be arranged in a manner which allows free circulation of space. The materials should to be sterilized should be perfectly dry. Test tubes, flasks, etc. should be fitted with cotton plugs. Petri dishes and pipettes should be wrapped in paper. Rubber materials and inflatable materials should not be kept outside, and it should not be kept inside. Sorry, the oven must be allowed to cool for 2 hours before opening, since glassware may suddenly break due to sudden cooling. The uses of hot air oven are as follows: to sterilize glass syringes, petri dishes, pipettes, and test tubes; to sterilize instruments like scalpels, scissors, forceps, etc., and also liquid paraffin, wax, and fats. To know whether the sterilization is complete or not, we can use the sterilization controls, such as spores of Bacillus subspecies niger, thermocouples, Brown's tube, which is a tube containing red color solution when inserted in each load, and a color change from red to green indicates proper sterilization. Moist heat sterilization, it is of three forms: temperature below 100 degrees, at a temperature of 100 degrees, temperature above 100 degrees. Temperatures below 100 degrees are pasteurization, vaccine bath, water bath, incipation, and low temperature steam formaldehyde sterilization. Pasteurization, which was described by Louis Pasteur, is a method of sterilizing milk, and it has two methods of doing it. It is a Holder method and a flash method. Holder method is 63 degrees heating for about 30 minutes, followed by rapid cooling to 13 degrees or lower. Flash method is 72 degrees for about 15 to 20 seconds, followed by rapid cooling to 13 degrees or lower. The dairy industry sometimes also use ultra high temperature sterilization, which is 140 to 150 degrees for 1 to 3 seconds, followed by rapid cooling to 13 degrees or lower. All non-sporing pathogens such as myobacteria, brucella, and salmonella are destroyed by these processes. Coxiella burnetii is relatively heat resistant and may not be sterilized by Holder's method. This is a principle of pasteurization, which is shown in a diagrammatic representation. Vaccines bath. Vaccines prepared from non-sporing bacteria may be inactivated in a water bath at 60 degrees for 1 hour. Water bath, which is in water bath, it is for 1 hour at 56 degrees on several successive days. Incipation is usually done in media containing egg or serums such as Lowenstein-Jensen or Lofer serum slope, which are rendered sterile by heating at 80 to 85 degrees for 30 minutes on 3 consecutive days. Low temperature steam formaldehyde sterilization is used when it is less when the sterilization should be less than 100 degrees. In this method, it is heated at 75 degrees with formaldehyde. These are the pictorial photos of incubator and water bath. Temperature at 100 degrees, at 100 degrees or more than 100 degrees is boiling sterilization, steam sterilization. Boiling at 100 degrees for 10 to 30 minutes kills all vegetative bacteria and some bacterial spores. Sporing bacteria requires prolonged periods of boiling. It is not recommended for sterilization of surgical instruments. Addition of 2% sodium bicarbonate may promote sterilization. It is used in disinfection of medical and surgical equipments. Sterility is not essential in emergency or fears under some conditions. Free steam at 100 degrees at normal atmospheric pressure. It is used to sterilize heat labile culture media. Cox or Arnold steam sterilizers are used. It consists of a vertical metal cylinder with removable conical lid. Single exposure to steam for about 90 minutes ensures complete sterilization. Sterilization is exposure of steam at 100 degrees for about 20 minutes on 3 consecutive days. The instrument used is Cox or Arnold steam sterilizer again. Its principle is exposure of all the vegetative forms, and in regular intervals heating kills the remaining spores, which germinates into vegetative forms, which are subsequently killed on next round of heating. It is used for sterilization of egg, serum, or sugar containing media. This is a photo of Koch or Arnold steam sterilizer. Temperature above 100 degrees. Steam under pressure. It provides greater lethal action of moist heat. It is quicker in heating up the exposed articles. It is easily penetratable in porous materials, and also the steam meets the cooler surface of the metals or the containers, and it condenses it to a small volume of water and produces considerable more latent heat. The autoclave is a gold standard used in sterilization, especially in India. In it, the water boils at vapor pressure, which is equal to that of the surrounding atmospheric pressure. When the pressure inside a closed vessel increases, the temperature at which water boils also increases. Saturated steam has penetrative power and is a better sterilizing agent than dry heat. Steam condenses to water and gives up its latent heat to the surface, which, when it comes to a contact with a cooler surface, the large reduction in volume sucks in more steam, and the process continues till the temperature of the material is raised to that of the steam. The components of autoclave, it consists of vertical or horizontal cylinder of gunmetal or stainless steel. The lid is fastened by screw clamps and rendered airtight by an asbestos washer. Lid bears a discharge tap for air and steam, a pressure gauge, and a safety valve. The holding period is generally 121 degrees for about 15 minutes under 15 lbs or psi of pressure. This is the picture of a sterilizer and the process it goes through. The operating valves, the pressure gauge, the safety gauge, and the way the air and the steam travels. This is a sterile autoclave. All the air from the autoclave must be removed. The admixture of air with steam results in low temperature being achieved, which results in which hinders the sterilization process. Materials should be arranged in such a manner which ensures free circulation of steam inside the chamber. Lid should not open or should not be open until inside pressure reaches the atmospheric pressure. Uses: to sterilize culture media, rubber materials, gowns, dressings, gloves, instruments, and pharmaceutical products. For all materials that are water containing, permeable, or wettable, and not liable to be damaged by the process. Useful for materials which cannot withstand the high temperature of hot air oven. Sterilization controls for this are thermocouples, bacterial spores, Bacillus stereothermophilus, Brown's tube, autoclave tapes. Filtration is also a method of sterilization, in which the solutions that may be damaged or denatured by high temperatures are used for sterilization of heat, labile materials such as sera, sugar solutions, and antibiotics. Types of filters are earthenware filters, asbestos disc filters, sintered glass filters, membrane filters, syringe filters, and air filters. Earthenware filters are manufactured in several different grades of porosity, used and basically used for purification of water for industrial and in drinking purposes. There are two types of earthenware filters, like unglazed or compressed diatomaceous earthen filters. In unglazed ceramic filters, there are Chamberlain and Dalton filters, and in compressed diatomaceous earth filters are Bergfield and Mandel filters. Earthenware filters are generally used in as candles in the filter which was being used uh, around in 2000s and 2010s. Asbestos filters are made of disc of asbestos, which are also available in different grades of porosity. It is supported on a perforated metal disc within a metal funnel. It is then fitted onto a sterile flask through a silicone rubber bone. The fluid to be sterilized is put into the funnel and it is connected to the exhaust pump through its tube. Asbestos filter holders looks like this. This is sterile flask, and there's a hole to pump the vacuum. Sintered glass filters, prepared by fusing finely powdered glass particles, available in different pore sizes. Pore sizes can be controlled by general particle size of the glass powder. The filters are easily clean, have low absorption properties, and do not shed particles, but they are fragile and relatively expensive. This is a sintered glass filter. Membrane filters. made of variety of polymeric materials such as cellulose nitrate, cellulose diacetate, polycarbonate, and polyester. Membrane filters are available in pore sizes of 0.015 to 12 micrometers. The 2 micrometer filter is most commonly used because the pore size is smaller than that of bacteria. These are routinely used in water analysis, bacterial counts of water, sterility testing, and for the preparation in parenteral uses. They are the membrane filters. They are here it is seen untreated, treated, and bottled water. Syringe filters. Syringes fitted with membrane filters of different pore sizes are available. For sterilization, the fluid is forced through the disc by pressing the piston of the syringe. Air filters. These are sterilized. Air also can be sterilized by filtration. Large volumes of air may be rapidly freed from infection by passage through high efficiency particulate air filters. They are used in laminar airflow system in microbiology in laboratories. The high efficiency particulate air filters can remove particles of zero point 0.3 micrometers or larger. There are two types of radiation used for sterilization. They are non-ionizing and ionizing. Non-ionizing radiations include infrared and ultraviolet radiations. Infrared is used for rapid mass sterilization of syringes and catheters. UV radiation with wavelength of 240 and 240 to 280 nanometers have shown bactericidal activity. It acts by denaturation of bacterial protein and interference with DNA replication. They can penetrate only a few into liquids and not at all into solids. UV radiation is used for disinfecting enclosed areas such as bacteriological laboratories, inoculation hoods, laminar flow, and operation theaters. Most vegetative bacteria are susceptible, but spores are highly resistant. Susceptibility of viruses is variable. Source of UV radiation must be shielded onto must be shielded others and to prevent damage to skin and eyes. Ionizing radiations include X-rays, gamma rays, and cosmic rays. They possess high penetrative power and are highly lethal to all cells, including bacteria. They damage DNA by various mechanisms. Gamma radiations are used for sterilization of disposable items such as plastic syringes, swabs, culture plates, cannulas, catheters, etc. Since there is no appreciable increase in the temperature, this is known as a cold sterilization. Large commercial plants use gamma radiation emitted from a radioactive element, usually cobalt-60. The advantage of this method includes speed and high penetrating power. It can sterilize materials through outer packages and wrappings, which is a quite an efficient ability, which permits sterilization. Bacillus pumilus is used to test the efficiency of ionizing radiation. The chemical methods of sterilization are are generally antiseptics and disinfectants. An ideal antiseptic or disinfectant should be fast acting in presence of organic substances, be effective against all types of infectious agents without destroying tissue or acting as poison if ingested. Easily penetrable material to be disinfected without damaging slash discoloring it. Be easy to prepare, stable when exposed to light, heat, or other environmental factors. Be inexpensive, easy to obtain and use, and also not have an unpleasant odor. Chemical agents act in various ways. The main modes of action are protein coagulation, disruption of cell membrane, removal of free sulfhydryl groups, substrate competition. Factors that determine the potency of disinfectants are concentration of the substance, time of action, pH of the medium, temperature, nature of organism, presence of organic matter. Disinfectants can be further divided into three groups, such as high-level disinfectants, intermediate-level disinfectants, low-level disinfectants. High-level disinfectants include glutaraldehyde, hydrogen peroxide, peracetic acid, and chlorine compounds. Intermediate-level disinfectants are alcohol, iodophores, and phenolic compounds. Low-level disinfectants include quaternary ammonium compounds. Alcohol, it is a most common disinfectant which is usually easy to avail, and ethanol and isopropanol are the most frequently used. used as skin antiseptics and act by denaturing bacterial proteins. It rapidly kills bacteria, including tubercle bacilli, but they have no sporicidal or virucidal activity. 60 to 70% is the most effective. Isopropyl alcohol is preferred to ethyl alcohol as it is a better fat solvent, more bactericidal and less volatile. Methyl alcohol is more effective against fungal spores. Aldehydes, two aldehydes, formal and glutaraldehyde, are currently used. Formaldehyde is an active agent against the amino group in the protein molecules. It is lethal to bacteria and their spores, viruses, and fungi. It is employed in the liquid and vapor states. A 10% aqueous solution is routinely used. It is used generally to sterilize bacterial vaccines. 10% formalin containing 0.5% sodium tetraborate is used to sterilize clean metal instruments. Formaldehyde gas is used for sterilizing instruments, heat sensitive catheters, and for fumigating wards, sick rooms, and laboratories. Glutaraldehyde has a similar action compared to formaldehyde, but it's more active and less toxic than it. It is used as a 2% buffer solution. It is available commercially as Sidex. It is used in cystoscopes, endoscopes, and bronchoscope sterilization. It is used for sterilizing also of endotracheal tubes, face mask, corrugated rubber anesthetic tubes, and metals. Phenol is obtained by distillation of coal tar between temperatures of 170 degrees and 270 degrees. It has a least lethal effect due to cell membrane damage. It has 1% phenol is enough for bactericidal action. Phenol derivatives like resol, chlorhexidine, and hexachlorophane are commonly used as antiseptics. Resols like Lysol is also commonly used for in soaps, cleaning floors, disinfection of excreta. Chlorhexidine, which is commonly known as Savlon, is used widely in wounds, preoperative disinfection of skin. More active against gram-positive than gram-negative bacteria. No action against tubercle bacilli or spores, and have very little activity against viruses. It has a good fungicidal activity. Halogens like chlorine and iodine are the most commonly used disinfectants. They are bactericidal and are effective against sporing bacteria and viruses. Chlorine is used in water supplies, swimming pools, food and dairy industries. Chlorine is used in the form of bleaching powder, sodium hypochlorite, and chloramine. Hypochlorites have a bactericidal, fungicidal, virucidal, and sporicidal action, which almost gives an all-around activity for it. Bleaching powder or hypochlorite solution are the most commonly used for HIV infected materials. Chloramines are used as antiseptics for dressing wounds. Iodine in aqueous and alcoholic solution is used as a skin disinfectant. 2% iodine is generally applied along with water ethanol solution of potassium iodide. It has an active bactericidal activity and a moderate action against spores. It is also active against the tubercle bacteria and viruses. Compounds of iodine with surface active agents known as iodophores are used in hospitals for preoperative skin degerming. Povidone iodine, which is commonly known as betadine, is used for wounds and also laboratory disinfections and also in skin preparations. Dyes such as aniline and acridine dyes are commonly used in skin and wound antiseptics. Aniline dyes includes crystal violet, brilliant green, malachite green, and are more active against gram-positive bacteria. They have no activity against tubercle bacilli, and they interfere with the synthesis of peptidoglycan of the cell wall. Their activity is inhibited by organic materials such as pus. Acridine dyes also are more active against gram-positive bacteria. Acridine dyes are very less in presence of spores. More important dyes, acrylic dyes are proflavin, acriflavin, euflavin, and aminacurate. They interfere with the synthesis of nucleic acids and proteins in bacterial cells. The metallic salts such as silver, copper, and mercury are also used as disinfectants. Proteins coagulate and have the capacity to combine with free sulfhydryl groups. These organic compounds, thiomycel, phenylmercury, and mercurochrome, are less toxic and are used as bactericides. Copper salts are used as fungicides. Surface active agents can be anionic, cationic, non-ionic, or amphoteric. Anionic agents are common soaps which have strong detergent but weak antimicrobial properties. Cationic act on phosphate group of the cell membranes like quaternary ammonium compounds such as benzalkonium chloride and acetyl trimethyl ammonium bromide. Amphoteric agents are usually take-over compounds. Vapor phase disinfectants are formaldehyde gas, which is employed for fumigation of heat-sensitive equipment, um, anesthetic machine and baby incubators, operation theaters, wards, and laboratories. Formaldehyde gas is generated by adding 150 gram of KMNO4 to 280 ml of formalin for 1000 cubic feet of room volume. This reaction produces considerable heat and is heat resistant and should be used in heat resistant containers. Sterilization is achieved by condensation of gas on vapor surfaces. Water after completion of disinfection, the effect of irritant should be nullified by exposure to ammonia vapors. Ethylene oxide is also used as a surface disinfectant or an air disinfectant, which is a colorless liquid with a boiling point of 10.7 degrees. It is highly lethal to all kinds of microbes, including spores. It acts by alkylating the amino, carboxyl, hydroxyl, and sulfhydryl groups in protein molecules. In addition, it reacts with DNA and RNA also. Highly inflammable and in concentrations of uh, uh, three concentrations of more than 3%, it becomes more inflammable. By mixing it with inert gases such as CO2, its explosion can be eliminated. It is used for sterilizing plastic and rubber articles, respirators, heart-lung machines, sutures, dental equipments, and clothing. It is commercially used to sterilize disposable plastic syringes, petri dishes, etc. It has a high penetrating power and thus can sterilize pre-packaged materials. Bacillus globigi, a red pigmented variety of Bacillus, has been used as biological control for testing of sterilization. Beta propiolactin, it is a condensation product of ketane and formaldehyde. It has a boiling point of 163 degrees, has low penetrating power, but has a rapid action. For sterilization of biological products, 0.2% beta propiolactone is used, and it is capable of killing all microorganisms and is definitely active against viruses. It is used in liquid form and it is used to sterilize vessels and vaccine holding. Recently, we used vapor phase disinfectants are hydrogen peroxide and peracetic acid. Hydrogen peroxide is used to decontaminate biological safety cabinets. Peracetic acid, it is an oxidizing agent and one of the high level disinfectants. It is used in plasma sterilization. Testing of these disinfectants can be done by these five tests, which is minimum inhibitory concentration test, which involves assessing the minimum concentration of the disinfectant required to kill the total bacteria present in the certain volume. The radial Walker test, the Chick Martin test, the capacity test, and the in-use test. Sterilization of prions, mainly by dry heat, 360 degrees for 1 hour, moist heat, 134 to 138 degrees for 18 minutes, and it also can be done under pressure or not under pressure. Chemicals, 25% sodium hypochlorite for 1 hour, sensitive to household bleach, phenol, and other disinfectants. Thank you.